I'm Xin-Yuan Guan from Department of Clinical Oncology, the University of Hong Kong. So today I will present our recent work. Hepatocyte differentiation model reveals two subtypes of HCC with different oncofetal properties and therapeutic targets. Okay, as we know, HCC is one of the most common cancers in the world, and with extremely poor prognosis. And also, it is believed that the development of HCC is a multi-step process involving many cancer-related genes, and including a lot of oncogenes. So recently, we found some of critical transcriptional factors such as CHD1L was dynamically expressed during liver development and contributes to the liver cancer lineage reversion. Therefore, a systematic characterization of liver development landscape might be help us to better understanding liver carcinogenesis and identify some driver events. Okay, recently we established an in vitro hepatocyte differentiation model, which started from embryonic stem cells, which can be induced to endoderm cells, liver progenitor cells, and mature hepatocytes. So cells from these four developmental stages, as well as two clinical HCC samples, including tumor and non-tumor tissues, were collected for transcriptome sequencing analysis. So the lower panel shows examples of expression patterns of some genes, including some stemness-associated genes such as ALK4, SOX2, etc. Okay, here shows the morphology of the cells at different hepatocyte developmental stages. Middle panel shows the representative biomarkers, and the lower panel shows the immunofluorescence staining of these biomarkers at different hepatocyte developmental stage. Oh, okay, so genes which shows both reached their peak expressions at different liver development stages, and also significantly upregulated in tumor tissue were selected for further study. Okay, here is hierarchical clustering analysis found different gene expression pattern at different stages of hepatocyte differentiation, which can be divided into embryonic-like, liver progenitor-like, and hepatocyte-like groups. Okay, so expression of subtype specific signature genes belonging to several developmental signaling pathways were detected at different liver development stages. As shown in the upper panel, the activation of cell cycle, pluripotency, and Gli signaling pathways were associated with embryonic-like features, while the activation of extracellular matrix and Wnt and TGF-beta signaling pathways were associated with liver progenitor-like features. You can see here, this is the genes. This is between ES and endoderm. Okay. Okay, so in the panel A, we can see gene GO and network analysis found HCC could be grouped into two major subtypes according to the top-ranked pathways and the molecular functions. That's the embryonic-like tumors and liver progenitor-like tumors. So in panel B, IPA upstream activator analysis was used to identify the common transcriptional activators of the oncofetal genes isolated from the two subtypes of liver cancers. So transcriptional factors both gain high activation Z score and fold change in the tumor were regarded as potential oncogenic drivers. As E to F1 here, and the SMART3 got the highest score in embryonic-like tumors and liver progenitor-like tumors. So they are selected as the potential upstream activator of these two types respectively. Okay, here the upper panel shows the selection of top-rated genes, including 6 genes in embryonic-like tumors and 4 in the liver progenitor-like tumors, which were regulated by the oncogenic driver. Lower panel shows the validation of these genes in series of HCC cell lines. These genes were used as different oncofetal signatures. So based on these oncofetal signatures, HCC patients can be divided into 4 groups. Okay, embryonic-like, liver progenitor-like mixed signature double positives and hepatocyte-like double negative tumors. So survival analysis was performed for HCC patients with different oncofetal signatures in HKU HCC cohort and TCGA HCC cohort. The results found that patients characterized as with mixture signature consistently show significant poor prognosis in both Hong Kong U and TCGA HCC cohort. So to test whether inhibition of E2F1 or SMART3 could affect the subtype-specific developmental signatures pathways, there are small molecule inhibitors, HLM and SIS3 were used to treat the HCC cell lines with different oncofetal signatures. So in panel B, we found inhibition of E2F1 with HLM could significantly downregulate the embryonic-like signature genes. In panel C, we can see the inhibition of SMART3 with SIS3 could downregulate the liver progenitor-like signature genes. Okay, in the panel D, HCC cells with embryonic-like signature, liver progenitor-like signature, or mixed signature, that's the double positive, as I mentioned before, were treated with E2F1 and SMART3 inhibitor at indicated concentrations. The percentage of cell growth inhibition was detected. So in the E, you can see, panel E, you can see tumor organoids derived from primary HCC patients with different oncofetal properties were treated with E2F1 or SMART3 inhibitors. And their sensitivities to drug treatment were examined. The results showed that treatment of inhibitors could effectively inhibit the organoid formation in HCC with embryonic-like, liver progenitor-like, or mixed, double mixed signatures. So to further evaluate whether targeting the oncofetal drivers might be effective in HCC treatment, HCC cells with embryonic-like, liver progenitor-like, or mixed oncofetal properties were stably transfected with shRNA specific targeting E2F1 or SMAD3. So compared with control groups, functional analysis found that the targeting E2F or SMAD3 could dramatically inhibit cell growth, colony formation in soft agar, and cell migration and inversion. So furthermore, in vivo targeting the oncofetal drivers with either shRNA or small molecule inhibitor could dramatically inhibit the tumor growth in the neuron mice. You can see that this is the results. So in summary, we identify gene expression signatures and liver cancer subtypes through linking hepatocyte differentiation with liver tumorigenesis. So activation of specified developmental signaling pathways along different stages of hepatocyte differentiation reveal two major subtypes of liver cancer with different oncofetal properties. So targeting the master transcriptional activator significantly suppressed their subtype specific developmental signaling pathways and effectively eliminated their tumorigenicity, so which provides therapeutic strategy in HCC treatment. Okay, thank you very much. I stop here.
Thank you very much, Dr. Guan. Does anybody want to ask a question? Amedeo? So let me ask one or two questions. The first
one. It was very, very, very interesting. And in these two models, which seem to be a model of aggressive HCC, I didn't see any pathway linked with the angiogenesis or neoangiogenesis, which I, in a way, expected to find. Have you got any data about it?
Oh, okay, yeah. So we actually, as I mentioned, for the sequencing data, definitely many genes were detected, including maybe genes associated with angiogenesis. But for us, we're only focusing on few genes, using a few genes, 6 genes, a total 10 genes. We use it as a signature to divide these patients, all HCC patients, into maybe 4 subtypes. Okay, so maybe we not include many other genes. However, our sequencing data, of course, showing many biological behavior changed. Okay.
Dr. Guan, the hepatocyte is a large cell, is the largest cell in the body. So do you see any change of the size in your cultures? And you're from the embryonic one to the, pre-hepatocellular. Do you see any change of the shape, of the size?
Actually, these parts was done by my collaborators. Okay, they sent us some figures, but I'm not quite sure. Yeah, I'm good in cell culture. If I culture the cell, I think I will pay attention to this. Yeah.
So because they get larger and larger. So in embryonic life also, they just, and the second question is, did you have the chance to check the non-tumoral tissue? You studied tumors, so you did non-tumoral tissues, because the tumor develop from the hepatocyte and it become tumors. So there is, if you follow the pathway in the non-tumoral tissue.
Yes, actually in this study, 2 non-tumor tissue were included, for our sequencing analysis. Yes.
Okay, did you see a development there somewhere? Your pathways, you see in the tumor tissue, there is any change?
Yes, of course, a lot's change because the genes we selected, we have two criteria. One is it's upregulated in some of developmental stages, but not in non-tumor tissue. Second, it's also upregulated in tumor tissues. So that's the genes we're looking with.
Okay.
Yeah.
Thank you. Other questions?
Yeah, Professor Guan, I'm Chen Hongma from here.
Hi.
Yeah, hi. Very interesting. I'm wondering, do you have the chance to check the immune signature in your samples?
No, in this one, no, because they're using a cultured model. Okay, so we are not using the model, generated from say mouse or others. Yes.
Okay, thank you. Thank you.
Mm-hmm.
Thank you.